Welcome, everyone. My name is Susan DeBleek, and I'm a member of the HEAL Data Stewardship Group. Thank you all for joining us today for the 22nd installment of our Fresh Fair webinar series, Fairly Sharing HEAL Data, Maximizing the Power of Publication. Today's webinar will emphasize the importance of fair research publication in HEAL, along with best practices and examples of how to package data associated with a publication. The webinar will feature lightning talks from HEAL-funded investigators, describing their experiences maximizing the impact of their publications by sharing data. This will be followed by a demonstration of the HEAL data platform. The webinar will conclude with a live question and answer session. This webinar is being recorded and will be shared with all registrants. If at any point you have a question, please feel free to type it into the Q&A box you can see at the bottom of your Zoom screen, and we'll bring it up during our question and answer session. Next slide, please. We're so pleased to be joined by our presenters today. Anthony Juehne, Dr. Bruce Taylor, Dr. Songthip Ounpraseuth, Dr. Bhuiyan, and Phil Schumm. Anthony Juehne is a program officer for the HEAL data ecosystem as part of the NIH HEAL Initiative. Juehne has over 15 years of experience within the field of healthcare informatics. He has published best practices for conducting and reporting health research to enhance reproducibility, transparency, and accessibility. Dr. Bruce Taylor is a senior fellow with NORC at the University of Chicago in the Public Health Department. He has over 29 years of experience in applied research, field experiments, and statistical analysis. His work includes multi-method approaches using nationally representative household panels, organizational surveys with service providers, jails, and law enforcement, and the collection of survey and biospecimen data. He is a co-investigator for the Justice Community Opioid Innovation Network, Juehne, their methodology center, analyzing the effectiveness of the Juehne network's 11 clinical research centers testing medicated-assisted treatments for opioid use disorder. Dr. Songthip Ounpraseuth is a professor in the Department of Biostatistics at the University of Arkansas for Medical Sciences. His research interests include pediatric clinical trials, computational statistics, prediction error estimation, dimension reduction and classification, and selection bias. He is an investigator for the study Identifying Quality Indicators for Substance Use Disorders and co-author of the publication Eat, Sleep, Console, Approach or Usual Care for Neonatal Opioid Withdrawal. Dr. Bhuiyan is a postdoctoral fellow at the Renthal Lab at Harvard Medical School and Brigham and Women's Hospital. He studies the evolution of peripheral nervous system cell types. Prior to joining the Renthal Lab, he received his PhD in bioinformatics at the University of British Columbia under the supervision of Dr. Paul Pavlidis. And Phil Schumm. Phil Schumm is the Director of Biostatistics and Statistical Computing in the Center for Translational Data Science at the University of Chicago. He has served as technical director for numerous data coordinating centers, including the NIDDK-funded Inflammatory Bowel Disease Genetics Consortium and the International IBD Genetics Consortium, for which he has led development of a data commons. He is co-PI with Robert Grossman of the HEAL data platform and co-leads the Data and Analytics Support Core for the NIDA-funded Justice Community Opioid Innovation Network, Juehne. Next slide. Today's session marks a continuation of our webinar series about publishing research findings. Part 1, titled Advancing Reproducibility and Data Reuse in HEAL, Guidance for Sharing Data Associated with a Publication, took place on July 20th, 2023. Next. The objectives for today's webinar is we want to provide practical approaches to establish and maintain strong connections between publications and digital assets like data sets, code repositories, and supplementary materials. We also want to share real-world case studies and success stories where researchers have effectively implemented FAIR and/or open science principles in preparing their data sets for publications. So with that, and the next slide, I am happy to pass it over to Anthony Juehne to begin the presentation.
Good afternoon or good morning, everyone, and thank you so much for making the time to join us today. And thank you to all of my fellow presenters and for everybody in the background that works so diligently to make these webinars happen. And we are so grateful that you all made the time to join us today. I want to start first with a very quick analogy that I think would be helpful in framing kind of some level setting and our objectives for today. So first, let's think of a scientific publication as the face of a watch. Most research publications also usually focus on reporting the impact of results, i.e. how this research progresses or moves the hands of a domain forward, a question or a practice forward, and its impact on where the current science is now. However, often the movement which is driving these hands and the structure, pieces, and operation of this movement is often opaque, ambiguous, or non-existent in reporting of research methods, thus leading to limited research transparency and accessibility and reproducibility. As we discussed in the first version of this webinar, which I encourage you all to revisit, as well as all the past webinars throughout the Fresh Fair series that provide excellent overview and insights into everything we're doing in the HEAL data ecosystem. The watch face can be easily read and tells the current state of a domain, a question, or a method, and where things are moving forward. However, the research data lifecycle and how the science is done in the background is often far more complicated. If one of those pieces break, the entire watch can break. If one of the pieces is not finely tuned, it will not run correctly, i.e. the entire method will not be able to be reproduced. Each member of your team is an active collaborator in making this watch work and provides subject matter expertise to the operation of each mechanism. If you're only sharing the face of your watch, you're not getting the full credit for all of your excellent work, nor is the community getting the most value out of the work you've all done. There are multiple frameworks for fair data sharing to enhance the reproducibility and rigor of research, all of which are exceptionally nuanced and vetted by leading experts in their respective fields. Many are tailored for a particular methodology, a particular data type, or domain of research. We'll share a library of resources in our slides and follow-up. And while I urge you all to explore the frameworks of highest importance for your particular research, we want to highlight some common themes across these frameworks to provide some easy to leverage guidance and calls to action so you can easily align with and participate in communities of open science. Science is complicated, and thus reproducibility is complicated, but we can make this easier. This is our goal. This is what we envision. So how can we operationalize such reporting and data packaging practices within a publication? The figure on the left is complicated, so you need not absorb every detail at the moment. The important point is this publication represents an example of how each piece of the scientific method is documented. All dependencies between each processes are clarified, from resources and protocols facilitating data collection to dependencies in the analyses pipeline. Both the data and analyses materials for reproducibility of their results are made reliably accessible through machine actionable persistent identifiers and as broadly available as ethically possible. In addition to example of how fair data sharing can be implemented when disseminating an imaging study, this publication also represents the value to the investigators themselves. For instance, this publication and the pre-publication have been cited over 160 times, and the GitHub containing the analysis materials have been forked 12 times. 12 times. That's a lot of collaboration and a lot of spread of this research impacts. Publications like this one, as well as the ones you'll hear in our lightning talks today from our excellent panelists, demonstrate our goal and provide a vision for how we can share science more openly. HEAL has carefully considered these objectives within our publication open access policy to support the broadest research impact and align with both trans NIH policies and initiatives coming from the White House. Let's go through a quick refresher of some of the policy directives. We ask that you deposit electronic copies of your publications in PubMed Central. Publish under a Creative Commons Attribution generic license, such as the CC BY 4.0 or an equivalent. Make your publications publicly available immediately without any embargo period. And finally, share the primary data for the publications through an appropriate HEAL compliant repository. Note, we have many resources out guiding you in how to share sensitive and controlled access data, and we also have controlled access repositories within the HEAL compliant data repository list and selection guidance so that you can make certain that you're aligning with the best practices for control and sensitivities of your data as well, and make it as openly and broadly available as ethically and sensitively possible. We also ask that we've also carefully considered this open science objective in our data sharing policy and provided resources and guidance to make compliance easy and efficient. We ask that you share data at the end of a project period or at the time of publication, whichever comes first. Deposit your data and materials in a HEAL compliant repository, as I mentioned, and ensure your data is reusable through creating and submitting your relevant metadata. Also, we've created a GitHub project materials template that you can easily copy and fork and leverage as a template for sharing your related tools, software, and code. We really want to ensure we're making this as easy as possible for you all, so we are all ears on questions and happy to provide guidance as possible. This slide presents some easy to leverage templates for data availability statements and citations. A few points to pull out here. Include a citation of the data in your reference list in data availability statements, such as the citation at the very bottom of the screen. Include the appropriate identifiers, such as the highlighted DOIs or accession numbers for your study's data collection at the repository where it is stored. If your study is registered in the HEAL platform, make sure to cite both the HEAL data platform study page and the repository where your data is shared, such as in the section containing both the HEAL data platform citation and the ICPSR citation. Remember to share all necessary metadata to support reuse of your data, such as your data dictionary and study protocol, and leverage appropriate standards. We have a lot of guidance and resources out that we are happy to point you to and we'll share within our slides today, which we'll share and follow up. Reproducing the stepwise methods of data transformation and analysis, which translate raw data into interpretable scientific results, is dependent on a computational environment and software. Thus, sharing solely the raw or final data is not sufficient for reproducible research, and the materials operationalizing the translation of raw data to final scientific results ought to also be shared. We've shared a study materials template within our GitHub, which you can copy to deposit your code and software and associated READMEs and metadata. But a few questions to consider here is, are all relevant metadata and documentation available for reproducing the analysis reporting in your publication? And are all materials available in a format interoperable with open source software? Lots to consider here, but we want to make this as easy as possible for you all. It's understandable that you may have some hesitancy, and there's been a lot of change in this space over the last decade. The stewards have shared some resources here that can assuage some of these concerns that you might have, but in general, the risk to getting scooped can be greatly mitigated. In fact, making your data fair will increase attribution and allow for appropriate licensing and citation, as we demonstrated in the publication above, and as you'll hear more today. Your data can be of high value, and you don't know the extent of value if you don't share it. Secondary analysis is now more important than ever, as you'll hear more about today, and sharing your data supports this. Data sharing is hard, yes, but we are trying to make it as easy as possible through a number of different resources we're making available. And you'll hear more how you can make it easy for yourself. Importantly, the benefits greatly outweigh the risks, especially when we're all facing the epidemic of chronic pain and addiction that have touched so many vulnerable lives and loved ones. And considering those internal benefits, sharing the data and analysis materials with your publication make your research more reproducible and presents many benefits to you and your team, including increased citation, collaboration, and attribution of your work, increased trust in findings through more efficient detection of errors and prevention of publication retraction or corrections. And internally to your own teams, it increased the handoff and teaming in your lab, as well as more efficient training by embedding their best practices into the culture of your labs and teams. And each team member can easily verify, test, and build upon the work of one another. We certainly hope that you're able to embed this and get as much benefit from your research as possible, and we want to make that as easy as possible for you all. In essence, through surfacing just a few identifiers within your publication linking to the related data and digital assets, we can produce valuable connections across the scientific landscape, driving further collaboration, institutional development, and unveiling areas of promising discovery and implementation. The graph here demonstrates how these resources can be easily connected to unveil those sorts of connections that you all are interested in exploring, and lots of promising areas for growth. Surfacing these resources will also allow them to be easily pulled into the HEAL data ecosystem. The HEAL semantic search will allow teams to find connections between biomedical concepts and gaps in the field, prime for promising discovery, preclinical to clinical translation, broad implementation, and clinical decision support. Through this discovery, translation, and implementation, we can accelerate solutions to addiction, overdose, and chronic pain. And as you'll hear more today, the HEAL platform will allow users to find studies and associated publications, easily access the digital assets associated with those publications, and pull them into computational workspaces wherein they can be reused and potentially harmonized with data from other studies, thus providing greater generalization of results, testing it for very important areas for clinical decision support, and in general, really just driving discovery and implementation in areas of growth in the research that you all are crucial experts in. One initiative I really love to highlight and encourage you all to get engaged with as well is the Litcoin initiative, which is aiming to build machine-readable, AI-ready knowledge by parsing the free text of scientific literature and detecting biomedical concepts and ontologies and important information within those publications. If you're really interested in learning more, I urge you to engage with Tyler Beck and Christine Colvis, as they're eager to engage with investigators that can share their publications and support in validating some of the AI-driven curation and detection of biomedical concepts within that scientific literature. It's a very, very important initiative that we as HEAL are happy to be partnering with, and we urge you all to get involved and learn more. Before I transition, I really just want to put out some very quick calls to action to you all so that you can easily get involved with us and easily participate in open and reproducible sciences as quickly and easily as possible. First, I urge you to revisit the checklist that the stewards have produced that demonstrates checklists for complying with the HEAL data sharing and open access policy. It's very easy to leverage and has a lot of important resources within there. I urge you, if you're a HEAL investigator, to register your study in the HEAL data platform. And through the resources that we have and the tools that we have, share the study level metadata and variable level metadata that you are producing within your study, or share your data dictionary directly with the HEAL stewards at the email you see there. Report your publications if you have one already published or upcoming through peer review to the email that you see here through healquestions@od.nih.gov. Link data and associated digital assets in your HEAL study page within your data availability statement. For instance, just to repeat, deposit your data in HEAL compliant repo to surface them in the platform, and leverage the GitHub template for your related digital assets, software, code, and protocols. We're going to be disseminating a feedback form after the webinar, and we urge you all to provide your feedback, questions, thoughts, concerns, suggestions, and we love to hear from you all as well. Share your DOIs and citations for your current HEAL publications so that we can get you all involved. The use cases you'll hear today present that a few steps can greatly enhance the value of your data and the value of your research publication. And I hope that you all are you all learn more today, and encourage you all to get to ask questions, thoughts, concerns, and look forward to hearing from you all. And with that, I will pass it over to Dr. Bhuiyan.
Hi, I'm Shams. As we get started, I just want to thank the organizers for inviting me and including me on what I consider a very important initiative. Next slide, please. Without good data sharing, I don't think that this project that I'm about to present would have been possible. And so without further ado, I'll present our work on the harmonized cross-species cell atlases, the trigeminal and dorsal root ganglia. So given the diversity of neuronal and non-neuronal cell types, the field has exploded in recent years with single nuclei and single cell studies to characterize the cell types found in these ganglia. These studies have provided important insight into ganglia evolution, ganglia development, and ganglia response to injury. And importantly, next slide, please. They have provided a ton of single cell and single nuclei data that we in the field can use to identify novel targets for pain therapeutic development. Next slide. So one great thing is most of these studies have provided their raw and processed data on Geo. However, it still remains challenging to compare data and conclusions between these studies because of the differences that exist between data sets. One difference is the fact that each study uses their own nomenclature to annotate cell types. And while this is a small point, it still adds confusion to comparing the results between studies. Additionally, technical differences such as differences in species sequenced, cells versus nuclei prep, bioinformatics pipeline, sequencing pipeline, as well as sequencing, sequencing platform can create differences in the number of cell types reported between studies, as I'm displaying on the right-hand side, and can also create data sets with low transcriptomic coverage. So our solution to these challenges was to integrate previously published single cell and single nuclei studies into harmonized somatosensory cell atlases for the field to use to identify cell type specific molecular features that one could potentially use to identify therapeutic targets. Next slide, please. For the purposes of generating a harmonized DRG and TG atlas, we took advantage of pre-existing tools. Briefly, we integrated 30 studies. We integrated data from 30 dorsal root ganglia or trigeminal ganglia studies using Seurat, the Seurat CCA approach, and constructed a DRG neuronal atlas, a DRG non-neuronal atlas, a TG neuronal atlas, a TG non-neuronal atlas. Next animation on this slide, please. Additionally, we reconstructed all 4 atlases using 5 other computational approaches. And we found that while we were presenting our final atlas using Seurat CCA, our findings were our conclusions from our final atlases were consistent regardless of which computational tool you use to construct the atlas. Where differences existed, next slide, please. Where differences existed, we found where in cases where the sequenced cell had a poor number of genes sequenced, and we found that reproducibly across the different atlases. Next slide, please. So here I'm displaying the harmonized DRG neuronal atlas. Each dot on this UMAP projection represents one cell or nuclei from 21 studies or 6 species. In total, across nearly 76,000 cells and nuclei, we observed 18 neuronal cell types. And these cell types were labeled by the marker genes that they expressed. And so, for example, the SST cell type we have up there is where cells and nuclei that expressed SST. Next slide. So you'll notice that we named all of these cell types based on the marker genes that they express. And as I mentioned earlier, each single cell study uses their own nomenclature to annotate cell types. Nomenclature can be based on marker genes, as we've done, but also on cell type function or even just a cluster number. So here I'm showing a bar plot that displays the percentage of cells or nuclei from individual studies that contribute to the calc-A SSTR2 cell type in the atlas. Above the bar is the nomenclature used by that study. So what we're calling calc-A SSTR2 is what Sharma et al. Called CGRP alpha, or what Wang et al. Referred to as ZCCHC12 SSTR2, or what Renthal et al. Referred to as PEP1. All are valid, but it does make comparing results between studies confusing. So next part of this. So to tackle this problem, we have set up the harmonized atlas to allow for rapid nomenclature conversion, to allow for rapid nomenclature conversion between studies. And in doing so, we believe that we have helped the field tackle this annoying problem, and it will improve people's ability to compare results across studies. Next slide, please. Also, one thing we've noticed when we constructed this cross-species atlas was that there is now 9 to 10 times more mouse data than there are human data. And in order to make a better comparisons between humans and the model organisms that we use, we really need more human data. Fortunately, to facilitate characterizing human cell types, 4 new pain centers have emerged in the US that aims to sequence a ton of new human data. However, a key challenge the field faces is that human ganglia tend to have a limited number of neurons. Next slide, please. And so what we found by anchoring to the harmonized reference atlas is that we can improve the atlas, the number of cell types identified in individual studies by almost 2 times great. We can increase it 2-fold. Next animation on the slide, please. And the code to anchor to our reference atlas is in fact available currently on our Git repo. Next slide, please. And I'll actually just jump to the next slide for the sake of time. One more, 2 more clicks. Yeah, just the next slide. And yeah, just before I wrap up and thank everyone, our resource is currently under revision. The processed and raw data for this will be available on Geo, but currently available is the web atlas to explore this repository. To explore this resource. So feel free to log on and check out your favorite gene and the cell type specific gene expression pattern. Thank you. And yeah, with that, I'd like to thank everyone for listening, as well as the Pain Precision Center leaders, members of my lab, and our collaborators. Oh, and I'd like to pass the mic on to Song.
Thank you, and good afternoon. Thank you for the opportunity to share our experience with our data sharing process. Next slide, please. So in the US, at least one infant is diagnosed with NOWS every 18 minutes. So obviously this constitutes a significant public health concern, mainly due to prolonged hospital stay, and many of these infants with NOWS are treated within the NICU. So in 2016, experts from across the field joined to help identify knowledge gap and increase research opportunities. And then shortly thereafter, the NIH HEAL Initiative brought together 2 networks, the Ideal States Pediatric Clinical Trials Network, in collaborations with the Neonatal Research Network, and 3 data coordinating centers that were uniquely poised to address some of these gaps. The NIH ISPCN is composed mainly of academic and community-based health centers from rural and underserved states, whereas the NRN composed mainly of academic health centers from urban states who manage primarily urban infants now. Next slide, please. So this collaboration led to the ESC NOWS trial, which had basically the goal of comparing 2 modalities and 2 methods of assessing infants with NOW. The usual care is using the Finnegan scoring tool compared to the eat, sleeping, and consoling care approach. This study has 2 phases. There's an in-hospital portion that's done under waiver of consent, which was conducted between September 2020 through March of 2022. This was a step wedge cluster randomized design, utilizing 26 sites across 18 states. As noted already during the in-hospital phase of the study, all eligible infants were enrolled under waiver consent to assess the short-term outcomes. We enrolled a little over 1,300 infants, and over 500 nurses were trained to carry out the intervention. Next slide, please. So the primary and secondary results are kind of noted below here. When compared to usual care, the ESC care approach decreased time until infants were medically ready for discharge by almost 7 days. We also reduced the percentage of pharmacological treatment, difference of almost 32.5%. And really, there was no difference in our safety outcomes through early infancy. So in short, our results kind of provided strong evidence to support the use of ESC and the effective approach for caring for infants with NOWS. Next slide, please. Part 2 of the study is still ongoing. Here we are wanting to assess the intervention's impact on infant and family well-being and the development through 2 years of age. Anticipated completion date around the end of summer of this year. Obviously, still a lot of unanswered questions in the field. Next slide, please. So currently, the in-hospital portion of our data is being shared, made available to the public. Currently on the NICHD Data and Specimen Hub, better known as DASH. This platform is a centralized resource hub, which allows researchers the opportunity to share and access the identified data from various studies. Next slide, please. Some of the values we feel that, in terms of sharing our data. First, obviously, we're following NIH and HEAL data sharing policies. And again, as noted by Dr. Collins, 2 of the cornerstones of science advancements are rigor in the design and performing of scientific research. And secondly, the ability to reproduce biomedical research findings. So obviously, when results can be reproduced by multiple scientists or research teams, it validates the original results. Another positive that we feel it enhances the scientific value of our studies through broad data sharing, which has been stated already. Our original study investigators will be acknowledged with any new publications, obviously utilizing our data. We also feel that this enables or helps early stage investigators by supporting hypothesis generating or secondary analysis of existing data. Next slide, please. The diagram shown here kind of provides a nice summary of the DASH submission process. First 4 steps are obviously the responsibilities of the PI or the data coordinating center, and then followed by the DASH curation team takes over once the data has been submitted. So basically, the basic steps include obviously gathering of the data and study documentation, the identification of study data, and then generating or creating code books, making use of the DASH data preparation tool to kind of update your data. Next slide, please. So some of the requirements for submissions includes things like the institutional certification for sharing. So the submitting institution must attest to at least one of the following: that the study was conducted under an IRB determination of exemption or waiver of consent, or the study was conducted as part of some IRB approval protocol. There has to be a specific language describing the sharing of the original consent, or where the IRB has reviewed that the consent and the determination that all data can in fact be meet certain standards and can be shared as proposed. And then you have the de-identification methodology, kind of summarizing how the data was redacted or masked, and then obviously the de-identification methods that was used to de-identify the data. You have the DASH code book template. This template data dictionary basically captures the information about the data sets, the variables, and the coded values. Next slide, please. Here's kind of a portion of our ESC DASH code book, which again provides the documentation of our study measures and all variables and their corresponding values. DASH provides a code book template that is organized basically through different sheets. The first 2 sheets for the submitters it provides obviously just the data set and the variable information. And then one of the sheet contains reference information to support code book development. Next slide, please. So yeah, in publishing and that you need permission from NIH. There's an extensive set of online forms to complete and that guides kind of the iterative review process and hopefully the acceptance of the data. You must include obviously study protocol, your de-identification procedures, all data collection forms, meta description spreadsheets, data in accept format, and then the dash code book. Your data must be packaged using packaged up using the dash preparation tool. And of course, this is a process that is iterative and can take months. Next slide, please. And that is it. Thank you, and I'll hand the mic off to Bruce Taylor.
Okay. I'm going to talk about the Juehne project today, which and focusing on our surveys of households in the U.S. And if we go to the next slide, the study that we base our work on is using the AmeriSpeak panel, we conducted 10 national surveys looking at public opinions towards drug treatment and stigma towards opioid use disorder, and looking at various covariates of those variables. And I'll go through some of the details of that. The AmeriSpeak panel itself is a nationally representative address-based sample of 55,000 households, covering over 99% of the U.S. household population, with a non-zero probability of selection from an established national frame. And the contacts with participants are made by snail mail, phone, overnight express mailers, face-to-face to capture harder to reach participants, and in terms of drawing people into the sample. Next slide. The household recruitment rate for AmeriSpeak is 37%, which includes a second stage of recruitment for harder to reach participants, and a correction for non-response. We apply weights that adjust the data to not only the U.S. Census benchmarks, but also non-response. The AmeriSpeak panel does two surveys a month in English and Spanish. Incentives are between $4 to $20, depending on the length of the survey. So for the 10 surveys that I'm going to talk a little bit about today, our participant size for the surveys range from 1,000 to 6,500 panelists. And we started this work in February 2020, and our last survey was in January of this year. And with the panel of 55,000 people, we're also able to screen for uncommon behavior. So, for example, if the problem we're looking at is something like 10% of the population, we still would have 5,500 people in our survey to address the questions that we're interested in. Next slide, please. And some of the topics here are listed, and folks can look through it, just to mention a few of them. A big focus of ours has been stigma towards opioid use disorders. We've also looked at stigma towards other drugs like crack cocaine, and also methamphetamines. We've also looked at mental illness, stigma, internalized stigma. We have a range of sort of policy that we've looked at in terms of Americans' attitudes towards that. We've also, a big feature of our study, because we're the Justice Community Opioid Innovation Network, we've also looked at criminal justice involvement for personnel. So looking at what role personal incarceration may have had, and also support from family and friends, and whether they've also might have been incarcerated at some point. We have extensive questions that we ask also on drug use, and that's covered here. Next slide, please. We've also had a series of questions during COVID-19, looking at COVID-19 related illnesses, looking at stigma towards COVID, looking at also vaccination and attitudes, and taking protective behaviors. We had several papers on that we've done. Another key construct that we've looked at in many of the papers is looking at the role of racism, structural racism, and making a difference in some of these constructs. And then we also collect a series of demographics, for our sample, including merging in external data at various levels of geography, because we do have the residence of each of the participants, and we're able to look at that. If we can go to the next slide, please. So some of the topics we covered we've had up to now, actually probably not, I think, on 21 papers, but we had a series of papers on substance use disorders and looking at stigma and differences in public opinion across various geographical and socio-demographic groups. So we've looked at a range of public attitudes towards substance use disorder. We've looked at individual, interpersonal, and neighborhood measures associated with opioid use stigma. We've looked at variables like a gender identity, stimulant drug use, and how that may change things if you've been using stimulants, and any internalized stigma you may be experiencing. Another big thing we've looked at is partisan media consumption as well, and we've seen, not too surprisingly, major differences depending on your political affiliation. And we've also looked at COVID-19 vaccine hesitancy, and in one paper we actually looked at the vaccine hesitancy and substance use stigma as well in the same paper. And we had a range of papers then just more broadly on COVID-19 pandemics. If we can go to the next slide, please. So in terms of data sharing and preparation, we've make our de-identified data available through the Juehne Data Commons, and Phil, after me, is going to talk about that. We've also had mechanisms for folks to get access to our data through a data use agreement. And we've also, in terms of the data that we provide a clean file with weights and computed variables for various indices that we have in the study. We've also, in terms of folks we've been working with, so we have each of the Juehne hubs we've worked with over the years, including the investigators from those studies and their postdocs, students, others who are clinicians we've worked with. We've also worked with folks with lived experience on these papers, and it's worked out nicely, we feel, like in terms of the collaborations and partnerships that we've put together. So in terms of the papers, most of them have been led by our team in the MARC, but also we've had quite a number of others from other hubs that have done this as well. Next slide, please. So we've gotten a lot, really, we found, out of data sharing with our papers, and really we've been able to really increase the use of the data to support open science tremendously through this sort of data sharing and working with all the different partners. We've really been able to sort of multiply the capabilities of our methodology center by being able to share our data and have others start working on the data. And we've had a sort of a variety of arrangements. In some cases, we've had other investigators come in and lead papers, or they may have joined us in our papers. And really, it's just become a shared responsibility just to get these results out there. And that's been a nice, satisfying feature of the Juehne work, is being able to get these papers out there, peer-reviewed, and get sort of people thinking about the results of them and trying to think about the implications for policy making. We've also done a video on the project on some of our findings as well, just making them more accessible to other audiences. And what we've really found is that through data sharing, we've gotten a more diverse group of researchers involved, including those who maybe haven't gotten funding that's been available through Juehne by giving them access to our data. Now they have, we think, in some cases, rigorous data for really to work with. So there's been a lot of mentoring along the way with the data sharing and getting people some additional skills to do some of the work. One of the things we've also done to try to make the data sort of more accessible or increase greater use of the data is we collect information from our Juehne community on areas that they would like to see as surveys. So in some cases, they're collecting data from particular jails, and then we have data that speaks to the entire community where the jail may sit in. So we have data that's not only at the national level, but we've also collected enough cases, at least least where the J-coin projects are happening, to do state level estimates. And some of our hubs have found that to be particularly useful. And that basically covers us. Okay, next I'm going to pass it on to Phil.
Okay, thank you very much, Bruce. And I'm just going to take a few minutes to try and sort of tie together all the things that you've heard so far this morning with a specific example. Next slide, please. And so thanks to the hard work by Bruce and his team, the data that he was just describing are actually available on the HEAL platform. And so I'm just going to walk very briefly through with you how to find the study, how to, well, actually, yeah, okay, just a sec. I'm going to share my screen. Okay, so the slide before that was up for a second has the URL for the HEAL platform. It's just healdata.org. That's what I'm looking at here. And there's a lot to discover here. I urge you to spend some time exploring, but I'm going to hop right to the discovery page. You can see up at the top that I've logged in just to spare you having to watch me go through my two-factor authentication. But what I'm showing you right here is accessible regardless of whether people have authenticated or not. And so I'm going to look for the data that Bruce was just showing. There's several ways to do that, of course, but I'm going to just go ahead and use his last name or his name here. And this pulls up the stigma surveys that he was just talking about. And I can go ahead and click on this, and this will show me a page devoted to the study. There is a permanent link at the top in the right-hand corner that will always point to this study, so you can use that to refer people to it in the future. I'm going to go ahead and click on the data tab here. And what this shows essentially is there are a number of things, both metadata for the study, and then also the data for the study. And so I'm going to go ahead. So a lot of studies you will find in the platform do not have data yet. They haven't finished collecting it. They haven't submitted them to a repository. Many more of the studies will at least have provided some metadata. And as I said, although I have logged in, anyone without logging in is able to come and download those metadata. I'm going to go ahead and download some of the study level metadata and just open them up real quick so you can see what we're looking at here. These are some study level metadata. This simply provides a name and an abstract for the study. In some cases, studies have provided quite extensive study level metadata. And in some cases also even variable level metadata, and that will let you see exactly what variables they've collected even before you go ahead and request access to the data. So I just wanted to point that out to you. I'll hop back over and come back to that same discovery page. Oops, sorry. There's quite a bit that you could have used to find that study, of course. I'm going to come back to the data, and I'm going to go ahead and attempt to download the data. And I did this because I wanted to illustrate for you. So the HEAL data platform is a federated data mesh, meaning that it does not store any data, but it simply interoperates with the repositories where people have submitted their data to provide access to them from a single place. And to do that, you have to go ahead and authenticate to the repositories where those data live. Bruce mentioned the Jcoin Data Commons, that's where his data live. And so I'm going to go ahead and just authenticate to that. And what you see here will be very familiar to any of you who use online services. I'm going to go ahead and authenticate with this Google account. And this asks me if, or prompts me to authorize access to the data from that location on behalf of me within the HEAL platform. I'm going to go ahead and say yes. And you can see once I've done that, I've got 30 days, 29 days before I actually have to do that again, which is kind of nice. So now I'm going to come back one more time now that I've done that. And now you should see I can go ahead and download the data from Bruce's study. It's going to take just a moment to fetch the data from the remote repository and bundle them together. I'm not going to take the time now actually to show, and I can go ahead and download them, and there you go. Let me just go ahead and show. I don't think you can actually see this because of the way that I'm sharing here, but I'm going to take a look inside that data package. It contains data in formats for a variety of different packages, you know, SAS data, SPSS, R, and extensive metadata that go with the data. And so I'm going to go ahead and just open some of those metadata for the first wave of that stigma survey. And you can see here, this provides things like the names of the variables, descriptions of those variables, even what kinds of values to expect in them. And as I said before, in many cases, studies will provide this variable level metadata even before the data themselves are available. At any rate, this leaves me with data from his study in whatever format I choose to use, and I can use them locally. As you see, I just downloaded them, or I can actually move them into a platform workspace. I don't have time to show those right now, but if we can come back to the slide after this, I wanted to show you one more thing which sort of ties this all together. So this is a feature that we're currently working on deploying, so you won't see it quite yet. But up at the top of the study page, if you're the person who owns that study, you're going to have a button that says record publication that's outlined in red there. And if you go ahead and click on that, move to the next slide, please. What that gives you is a form where you can enter one or more publications that use the data that you have created with a DOI or PubMed ID. It also, importantly, allows you to provide other resources in addition to the data and the publication that you may want to link to this so that people can reproduce and extend your results. And so those could include things like a GitHub repository with the code or other kinds of repositories as well. And then the final slide, once you have done that, then people, when they visit your study page, will be able to see that list of publications on the study page. So as I said, those last 3 slides I showed is a feature we're currently working on developing and rolling out, but we'd be very keen on whatever kind of feedback folks in the audience may have on how we're doing that. And that wraps up what I have to say.
Wow, thank you so much, Phil, and our presenters for sharing. I just wanted to share our key takeaways, our practical strategies for establishing and maintaining linkages between research publications and digital assets like datasets, code repositories, and supplementary materials. We wanted to offer you all practical methods for connecting your research publications with those digital assets and also highlight the wealth of available resources. Next slide. We are asking that for publications, you align with relevant reporting guidelines and, of course, NIH policies, and deposit data and materials in HEAL compliant repositories. Next. And as you prepare for publication, oh, sorry, one more. Just consider, just wrapping up, as you prepare your data for publication, just consider what is needed to align with data packaging. So for this, we are going to jump into the question and answer session. Thanks to our attendees for already putting some questions in there. And I'm going to ask our speakers to turn on their videos. And we're also joined by Dr. Bhuiyan Smith. So we're going to stop screen sharing for a moment and jump right into our question and answer session. So Anthony, I wanted to hear about how HEAL enforces sharing HEAL initiatives funded research data only in the HEAL compliant repository list. So if you could speak to those repositories.
Yeah, thank you so much. An excellent question. So the stewards and platform team conducted an in-depth review of repositories across the scholarly landscape, leveraging a number of different leading standards such as Core Trust Seal, and looked at overall sustainability as well as some of their technical features to ensure that they were appropriate for both accepting, curating, and storing data from HEAL studies. And we want to make certain that we were gathering repositories that were both aligned with the needs for sustainability, as well as would accept all of the data that was being produced by very, very diverse landscape of research within HEAL. So all of those principles are available publicly, and also urge you to look at the HEAL compliant repository list. But also, if you have a particular repository that you're interested in depositing in, we do raise that to us and the stewards so that we can see if there are any needs for adjusting and including and extending that list to include that particular repository. We want to ensure that as we are building a federated architecture within the HEAL data platform, we are both being parsimonious in the repositories that we selected, as well as covering a broad landscape of all the data needs for HEAL investigators, and want to ensure that we are also aligned with those standards and principles. So the current landscape of repositories that we've collected and are on that list should cover all of the landscape of HEAL data, and a large swath of them will cover at least 85% of HEAL data. But we do urge you all to, if you have questions or concerns, do raise that to us.
Thank you. And then, Phil, is the HEAL data platform like the dataset catalog compiling HEAL initiative data only?
So primarily HEAL initiative data, although also HEAL adjacent data. So data sets that are, likely to be of interest to people working with HEAL data sets are definitely within our purview as well. The idea is to provide a singular place where somebody can pull data from multiple related sources and use them all together. The second part of that question, at the moment, we are working simply to interoperate with the repositories that are on the HEAL compliant list. Our ability to access data from other repositories, as you know, is dependent on those repositories having APIs that permit that kind of access. And so at the moment, we are focusing on the HEAL compliant repositories.
Thank you. And then for our lightning speakers and for Brian Smith, reflecting on your experiences, what pivotal guidance would you offer to an early stage investigator embarking on a similar journey, and particularly around data sharing?
I look forward any of the lightning speakers do raise. I think one of the pieces that I would urge you all is the earlier you start with thinking about data management and preparing your data for sharing, the earlier you think about that within your research lifecycle, the easier it's going to be downstream. And also do take a look at the resources that the stewards have shared on healdatafair.org, as well as a lot of the documentation and resources that the platform has, as it can make that process simpler.
Yeah, go for it, Song.
Yes, so I'll add to that as well. So I don't think this is only relevant to just early stage investigators, but just all researchers in general. And for the purpose of data sharing, when at all possible, it would be appropriate or good to have some sort of standardized code book. And part of the reason why I say that is that this allows for cataloging and describing and categorization of each of your variables submitted to the data repository, whether it be Dash or any other repositories, for other researchers to understand and accurately interpret the variables and the corresponding values of the study data. Also, standardization also allows for cross-study comparisons of the data sets that are shared to the same repository. And then finally, this also facilitates data submission process and helps with quality assurance.
And Shams, you were going to add to that?
Yeah, I generally echo what Song just said. I 100% agree. Just to add to it, I think I have benefited from when I keep good documentation of my code, both in code documentation and README files. And I think early on in my career, I suffered from not keeping good documentation. So I highly recommend documenting your code, because come publication time a year or two later, that's going to help you a lot and help people try and reproduce your work as well.
And then Bruce, do you want to close this out?
Sure. I was just going to add that I think it's important to consider contacting the investigators of the study. We get a lot of outreach from other researchers. I always find that helpful to give people a briefing of some of the ins and outs of the data. And I think you'll find most of the investigators are pretty willing to share and give you some good guidance. So, yes, thank you.
Wow, thank you so much for that. So I'm sorry it was such a quick Q&A. I'm going to jump back to the slides. And wanted to again thank our speakers. We're so happy to get to hear from you all today. And I also wanted to acknowledge the behind the scenes team listed here who works diligently to bring this webinar to production. So thanks to all. So next slide. Thanks to our audience for attending. We're really glad that you participated. In addition to receiving a recording of the webinar, we're also going to email everyone who is registered a list of resources. This slide deck will be shared, and a link to our Slack channel and Connect With Us form are going to be posted in the chat. We hope that you will reach out to the HEAL stewards for questions or support as you embark on your FAIR journey to maximize the value of HEAL data. So looking forward to seeing you all at our next webinar, April 18th at 1 p.m. Eastern, which we're going to be focused on variable level metadata. Thank you all again so much, and have a wonderful day, everyone.